to the lecture series in roles of nanomaterials in agriculture. So, we are into the animal production section. So, in the last class, if you recollect, we talked about the role of cerium oxide nanoparticles in changing the muscle mass, and as a matter of fact, there were 3-4 things what we gathered from it was that there is an increase in the muscle mass, there is increase in glycogen concentration, there is an increase in ATP concentration. So, in other words, these muscles are energetically more robust, and there is a increase in the type 1 fiber ratio, as well as there is an increase in the proteins or membrane proteins involved in electron transport chain across the mitochondria. While going through this, I did not mention about how really the muscle develop, in what context really these things changes. So, what we will do today is we will talk a little bit about the muscle. Now, that will kind of give you an idea that how really the whole biology works and how these nanomaterials makes a difference. Okay. So, if you look at any of the muscles very carefully, so if we have to, so since all these are dealt mostly with the muscle, so there are 3 ways you can classify the muscle, and we have only talked about one muscle, actually 2 different kind of muscles we have talked about. So, we talked about skeletal muscle yesterday in the previous class. Skeletal muscle, the muscle which are lining our gut or making our gut or the alimentary canal and everything, those are smooth muscle, and then you have cardiac muscle. So, we have talked about the repair of cardiac muscle and a positive effect of cerium oxide nanoparticle, okay, CO2 nanoparticle, and we have talked about a positive role of CO2 nanoparticle along with exercise in improving the skeletal muscle performance. So, essentially what happens is when the muscles are formed, so I will take you back to a bit of the development biology. When the muscles are formed in the mother's womb, the way it starts is there are these single cells which are destined to become muscle, any kind, it could be any kind of muscle, okay, because all the 3 muscle development is similar. These are those single cells, and these single cells are called myocytes. Myo means muscle, cytes means cells, okay, cells which are destined to become muscle cells. So, these cells over a period of time they form, they start to divide like this. So, each one of these masses starts to divide. Some finite number of divisions they go through, at a time. Something like this, followed by this division process. So, myocytes going through division, followed by this division process, they started to move and align like tubes. So, they align like this. This is very essential for you people to understand, because all the reference papers I will be giving, unless you understand this process, you won't be able to appreciate how those muscles are formed. That is the reason why we are taking this a special fragment of our course where we are talking about the muscle development. Okay. Something like this. Okay. These are long arrangement, long linear kind of arrangements which happens. So these cells are now migrating and aligning. Migration or migrating followed by aligning. As you could see here, they are aligned. And this is basically telling you the migration happening. Once they are aligning, there is an interesting process which happens. These cells started to lose their individual boundaries, and what they form is something like this. And all the nucleus, so these are the nucleus which are present. All the nucleus, so this is how the structure almost becomes like a tube. All the nucleus are arranged like this. A very typical of a skeletal muscle I am talking about. Okay. So these tubes are called myotubes. Okay, which is the smallest unit of muscle. Now these myotubes then several myotubes like this align with one another and form what we call as myofibers. Okay. So second level of alignment and formation of supramolecular assemblies formation of supramolecular assemblies, and these supramolecular assemblies are called myofibers. As you see, the word myo remains common. Now from site out here, it forms tubes out here, and then it forms fibers. So when I told you in the previous class there is a change in the fiber ratio, I essentially meant this fiber ratio. Now there is something else which I haven't mentioned as of now. That's what I am going to come. So this is your skeletal muscle development. Now there is something else which is happening. Now each one of these, so we know that these skeletal muscle generates force. If you recollect, when we talked about exercise as a double-edged sword, we talked about the ROS, reactive oxygen species, which is generated during exercise, is essential for force generation, right? So now coming back to this part, so who is generating the force? How the force generation is happening? Force generation, how it is happening, and who are the key players in force generation. Okay. Now when we look at these myocytes, Now, if I take a single myocyte here and enlarge form, okay. So this myocyte here is the nucleus. It has lot of these crisscross proteins in it. Coming to those, what are those? In its cytoplasm and along the periphery. Something like this, okay. And these proteins are termed as actin and myosin. These proteins are the smallest unit. These are the ones which are the key players in generation of force. How is it so? Now, these actin myosin filaments are randomly arranged inside the cytoplasm. Now, what happens when these cells divide? Again, they are randomly arranged. Then when they are migrating, it is perfectly fine. Then once they align, still they are randomly arranged, but then slowly there is a molecular superassembly happens where these actin myosin filaments almost arrange like this. Okay, something like this. Almost they align like tubes. So you have these cells, something like this. Now, once the myotube is formed, they follow this arrangement like this. Something like this. And there are discontinuities which you could see as something like this. So, if you see a muscle fiber, you will observe something like, we will come to that, what are those discontinuities are. It looks like discontinuities, actually those are not discontinuities, those are something like this, structures like this, which are also called Z lines. Now, what you observe, it's kind of, I'll go a little slowly for you to realize, these actin myosin initially were randomly arranged, okay. These were randomly arranged. Then while it is forming a supramolecular assembly out here, now actin plus myosin filaments forms a aligned tube like structure. This alignment and formation of tube like structure is one of the finest program arrangement of proteins which happens, and it is very, very unique to look at. And then once these individual cells loses their boundary and form something like syncytium, it's a continuous structure, something like this. This arrangement continues, and not only they form further rearrangement, okay. And it has been observed during development, while we are developing in mother's womb and about to come out. These myosin filaments changes their type. So, some of these myosins which are found while we human or any of the species which grows in their respective mother's womb have a different kind of myosin protein during their fetal stage. Okay, so they have a fetal myosin, and myosins are a heavy chain. Okay, so these are called fetal myosin heavy chains, and as they mature, as we are taking birth, they become much more mature, and there is a change in their protein conformation. They become more like mature, more like adult like myosin heavy chain. And they are also, sometime in biological literature you will come across this word called MHC, myosin heavy chain, type 1, type 2. Likewise, there is wide array of subtypes of myosins which you could see in the literature. So, during this development, as these tubes are forming, aligning, their myosin heavy chain type changes, and it is one of the most beautiful example of development. Okay, and there are cassettes of gene families which are involved in regulating this process. What is important for you to realize, this rearrangement of a structure, and this is where I am coming. If I take you back, whenever we talk about change in the fiber ratio because of the application of ferrium oxide nanoparticle, then we are talking about hitting upon the ratio of or type of myosin filaments. It is that deep where the impact is being observed, and that is why I am taking this extra step backward to teach you that you have to appreciate that this is not something ordinary. This is something out of the world to really look at. Wow, this is affecting all the way up to, without doing any kind of genetic manipulation. This is a simple, you are giving either in the diet or through blood or intravenous or subcutaneous into the muscle. If you give these kind of nanoparticles, which essentially is a simple redox couple which is there, like +3 to +4, sorry, +4 +3, okay, or +3 to +4, autocatalytic redox couple is bringing about changes which were never even thought of. It's something mesmerizing when you see the pattern with the thing, because this you will be able to appreciate better once I will provide you the relevant literature where you will see that changes are drastic, really drastic. I mean, that's something never ever people thought that without doing any kind of genetic manipulation or anything, they will see these kind of changes. Like, think of the change in these kind of, mostly these are iron sulfur proteins across the mitochondrial membrane, these ones. It's something phenomenal. So, now coming back where I was. So, it is out here, the fetal myosin heavy chain transformed into adult myosin heavy chain in these kind of muscle. Okay. Now, how this arrangement really looks like, that will be our next class. What we will be talking about is, now we have already talked about who are the players. Now, in the next class what we will do is, we will talk about the arrangement of actin, myosin filament, which are involved in force generation and role of mitochondria in this whole process, and this force generation will be governed by two basic biological processes: sliding filament theory and excitation contraction coupling apparatus. Okay, and the role of mitochondria: energy source, electron transport, and in this whole process, how our target sodium oxide redox couple plays a critical role. And next, what we will do, nano and microstructures to study force generation by muscle. And in this fragment, we will talk about nano and micro cantilevers and AFM. Atomic force microscopic tips. How these could come so very handy to study the force generation. So I'll close in here. We'll resume our journey on this part in the next 2 or 3 classes. Thank you.